Swayam Prabha. Digital India. Educated India.
Hi everybody, this is Dr. Ravindra Maradi from Department of Biochemistry, Kasturba Medical College, Manipal. In this lecture, I will be talking about overview of metabolism of sulfur containing amino acids. First thing, we should know what are sulfur containing amino acids. Out of 20 amino acids, 3 amino acids are sulfur containing amino acids. Those are cysteine, cysteine and methionine. So, the cysteine is a non-essential amino acids because you can synthesize it in our body, and this cysteine can be converted into pyruvate. Cysteine is nothing but a dimer of cysteine. Two cysteines are joined together to form cysteine, whereas methionine is an essential amino acid. That means you need to take this amino acid in your diet. So, our body cannot synthesize methionine, and this methionine is glucogenic in nature. That means the carbon skeleton of this methionine can be used to synthesize glucose, and if you will see in the metabolism of this methionine, this can be converted into succinyl CoA, and which can enter into TCA cycle, and from there you can synthesize glucose. So, degradation of methionine is equal to synthesis of cysteine. Cysteine you can synthesize during the metabolism of methionine. If methionine is not available, then cysteine becomes an essential amino acid. We will see the catabolism of how methionine is metabolized. First reaction is this methionine is converted into S-adenosyl methionine. Okay. If you see the naming, or if you see the structure of methionine, there is a difference in this adenosyl group. The adenosyl group is added to the methionine, and that binds to the SH group of the methionine. So, here you are transferring this adenosyl from ATP. ATP is the donor of this adenosine, and meanwhile, this ATP is converted into inorganic phosphate and pyrophosphate. So, the enzyme this pyrophosphate further can be hydrolyzed to form 2 inorganic phosphates. The enzyme required here is S-adenosyl methionine synthetase. So, as the name itself indicates, you are synthesizing S-adenosyl methionine from methionine by donating a adenosyl group to methionine. The next reaction, what happens, the methyl group carried by the methionine to the S-adenosyl methionine is donated to some of the methyl acceptors, and that methyl group is after getting that methyl group for some of the acceptors, they will be converted into methylated products. For example, epinephrine. Okay, if you donate during the synthesis of this epinephrine, the methyl group is donated and epinephrine can be synthesized, and even during creatine requires this methyl group for its synthesis. Then after donating removal of methyl group, you form S-adenosyl homocysteine. Okay. Here the methionine without the methyl group with the adenosyl group is S-adenosyl homocysteine, and the enzyme here is methyl transferase, because you are transferring a methyl group from S-adenosyl methionine to S-adenosyl homocysteine. In the next reaction, what happens, this S-adenosyl homocysteine remove that adenosine group, then you will get homocysteine. So, you are hydrolyzing here, the hydrolase enzyme hydrolyzes acetoacyl homocysteine to homocysteine. So, this is the important molecule in the metabolism of this methionine. So, till that this reaction, what is happening here? You added one adenosyl group to methionine to form acetoadenyl methionine, then a methyl group is taken out from methionine and again the adenosine is taken out. So, ultimately what is happening here is methionine to homocysteine, there is one difference is that methyl group is taken out from methionine, because ultimately you added the adenosine and taken out the adenosine, that does not make any difference to the structure, but you have taken out an extra methyl group that is present in the methionine to form homocysteine. Next, what happens is this homocysteine combines with the serine to form cystathionine. So, the enzyme required is cystathionine beta synthase, and this requires vitamin B6 for its activity. So, homocysteine plus serine forms cystathionine in the presence of vitamin B6, that is pyridoxal phosphate, to form cystathionine. The enzyme is cystathionine beta synthase. Then this cystathionine undergoes hydrolysis to form cysteine and alpha keto butyrate plus ammonia is produced here. So, serine is converted into cysteine and the remaining part of this homocysteine, that is converted into this alpha keto butyrate, and then that is converted to propenyl CoA. Propenyl CoA is converted into succinyl CoA and succinyl CoA enters into the TCA cycle. Then the enzyme required here is cystathionase. Cystathionase splits this cystathionine into cysteine and alpha keto butyrate plus ammonia, and this also requires vitamin B6, and desulfuration of cysteine forms the pyruvate. Okay. That is why we can call this methionine as a glucogenic amino acid, because you are forming this succinyl CoA and also the pyruvate. The other pathway this homocysteine can go into is direct conversion of homocysteine to methionine, and this reaction is very important. During its metabolism, thus methionine donated its methyl group for methylated products, some of the products like epinephrine and creatine, and if you give that methyl group back to homocysteine, you can synthesize the methionine, and this reaction is very important. You need to provide that methyl group, and that methyl donor is methyl cobalamin. Methyl cobalamin donates the methyl group and converts homocysteine into methionine, and you get the cobalamin, and again this the enzyme required here is methionine synthase, okay, or homocysteine methyl transferase is the other name of this enzyme, because you are transferring a methyl group to homocysteine and you are synthesizing methionine. And in this reaction you also require N5 methyl tetrahydrofolate, which is taken up by cobalamin to resynthesize methyl cobalamin, and this is very important, because if this methyl tetrahydrofolate this is if you do not take out this methyl group, the free tetrahydrofolate is not available if in case of B12 deficiency. So, the folate is trapped in the form of N5 methyl tetrahydrofolate. So, the cobalamin deficiency can cause deficiency of the folate deficiency, and folate free folate is not available for its activity. That is why this reaction is also very important in case of this folate trap. So, any block in the synthesis of or conversion of this homocysteine to this pathway, that is cystathionine, cysteine and pyruvate, that increases the blood homocysteine level, or the block here conversion of homocysteine to methionine. Either one will cause homocysteinemia, that is increased homocysteine level in the blood causing homocystinuria, and this is very very important because this homocysteine is associated with lot of disorders, like it is directly associated with coronary heart diseases, stroke and Marfan like habitus. A lot of things are associated with this increased homocysteine level. That is the about the overview of metabolism of this methionine. Just to summarize, we discussed about this methionine, and that methionine is converted into S-adenosyl methionine. Here is the donor of this adenosyl group from ATP, and then S-adenosyl methionine, there is a methylated reactions, that methyl group is taken out and forms S-adenosyl homocysteine, and that if you take out that adenosyl group from S-adenosyl homocysteine, you form homocysteine, and then homocysteine can enter into two pathways. One, it can convert into cystathionine, it is a B6 dependent reaction, and cystathionine can be again converted into cysteine, again it is a B6 dependent reaction, and it can convert into pyruvate, and one of the byproduct can be converted into propenyl CoA and succinyl CoA. And the other important reaction, direct conversion of homocysteine into methionine. Here you are adding a methyl group, methyl transferase or methionine synthase, and this requires two vitamins, vitamin B12, cobalamin and tetrahydrofolate. Any genetic deficiency of the enzymes, this is cystathionine beta synthase or methionine synthase, or any one of the vitamin deficiencies B6, B12 and tetrahydrofolate can cause homocysteinemia. I hope this lecture will help you to understand the metabolism of methionine. Thank you.